Hopefully you remember it from lab, and if not, hopefully you can retain it from here long enough to get you through today's lab. We're doing the dissection of the sheep brain today. Has anybody done it already? How was it?
That was fun.
Do you have any problems finding the stuff? Alright. So, Wilsey. So, most of y'all could probably come up with this list on some level, right? Homeostasis, it's got to take in sensations, got to figure out what they mean, got to make a response. A lot of our higher order functioning is going to come from this stuff as well, so that's pretty easy. One of the very basic questions is just going to be, can you identify some part of the brain, right? So we've got a transition from everything that's in here within the confines of the cranium. There's a hole here. What's the hole? Foramen magnum. Everything above the foramen magnum is brain. Everything below is spinal cord or something else, right? We're going to chop it up into major regions of the brain. All of this is technically brain now. We've got the big wrinkly mess that everybody associates with the brain, right? That's the cerebrum. That's what we're going to start with. And then we're going to jump down to the bottom, and then we're going to work our way up. We're going to go from the brain stem, right? And then we're going to talk diencephalon, and then we're going to come back here to the cerebellum, right? So, all right, so starting with the cerebrum, right? This is one you should know, right? We talked about this upstairs in the lab. This is going to separate one hemisphere of the cerebrum from the other. This is the longitudinal fissure. One of the things that you will do today, if you have not done the dissection yet, is you're going to take a big old knife or a scalpel and you're going to go right down the length of that longitudinal fissure and do a mid-sagittal cut separating one side from the other. The largest part of the human brain is comparatively large in us and not as large in other animals. This is where a lot of our higher order functions and a lot of our consciousness is coming from. Two hemispheres. They are not equal, right? We're going to lateralize some function to one side or to the other, right? We all heard of the left brain, right brain sort of thing. So some of that's real, some of it's not real. So the big division between them. The longitudinal fissure, right? So this is basically a dorsal view of the brain. Here we've done a coronal slice through it. I used this when we were talking meninges. This is one hemisphere. This is another. Right? You can see the meninges actually folding down in between them, including both the dura and the arachnoid mater. Now on Wednesday, none of my brains had the arachnoid mater and the dura mater still on them. Anybody today who's done it already? Did you also have meninges on them or not? Okay, so that's good and bad. It makes it easier, but it means you're less likely to see the cranial nerves, right? What cranial nerves did y'all find if you're not in my lab? Anybody?
Crickets.
All right. Did y'all find any cranial nerves? Okay. Now, that makes me a little nervous for y'all. So, we'll see. We got the wrinkly mess, right? The wrinkles are important, as it turns out. We've got a lot of the ridges, right? Ridges are gyri. We've got the grooves between the ridges, right? These are the sulci. Each and every one of these grooves, each and every one of these ridges has a name, right? That's unfortunate. The good news is that you really only have to know two ridges by name. And you really only need to know a couple of the grooves as well, right? So we'll deal with them when we come to them. This is, what kind of section is this? Good guess, but no. It is a coronal section. So, if you do a coronal section through this, right, you can see different regions. You can see the white and the gray matter, right? White matter here, gray matter here. Now, in a spinal cord, when looking at those models in the slides, where's the gray matter? Is it deep or superficial in the spinal cord?
Superficial.
Say it. Deep, right? And then the white matter in the spinal cord is superficial. We're going to flip-flop it when we talk about these structures within the brain. So for both the cerebrum and the cerebellum, the gray matter is on the outside in a region called the cortex, right? The white matter is on the inside. Let's see if we really know our stuff. What's this great big hole here? That is a ventricle. Which one?
Fourth one?
No.
Third. Second. First.
1 through 4.
Only 4.
Not the third. So it's got to be one of the laterals, right? We've got one lateral ventricle, the other lateral ventricle, right? So what's that hole there then? That's the third. You don't actually see the fourth here because it's in a section that this coronal slice doesn't go through. Right? You should also be able to do some of the lobes on this picture. What lobe of the brain would this be? Nope. Yeah, it'd be the temporal lobe. Right? You've redeemed yourself, Ray. Good job. So we've already mentioned this in the lab, but it didn't go into detail. Right? White matter, myelinated axons. Right? The gray matter, unmyelinated axons, but more importantly, the bodies of the neurons are usually located within the gray matter as well. Right? So what that means is that the more gray matter you have, the more neurons you have. The more neurons you have, right, the more connections you can make, the better able your brain is to function. Right? So what are the benefits of having a wrinkly mess? Since your gray matter is going in and out with the wrinkles, right? The wrinkles give you more neurons, essentially, right? It's that old same thing that we've talked about already, right? We said that with the connection between the dermis and the epidermis, right? It was like this because it increased surface area, right? You said you're going to see it in AMP2 when you talk about microvilli, right? Well, same phenomenon. Wrinkles give you greater surface area. So it's important, right? When the wrinkles aren't there, it doesn't even look like a brain, right? But that's what that is. That's a brain of somebody who suffers from something called lysoscephaly, right? They don't have the ridges and the grooves. They're gray matter, there's less of it, and the arrangement is incorrect, right? And so the result is that you impair functioning of the brain. Right? If you make it all the way to term, right, if you can actually have a viable birth of an individual with lysoscephaly, they don't end up living that long in any event, right? Because of cognitive and because of just general impairment of the functioning of the brain itself. So, all right, so we should have done this in the lab as well. This is one of the easy questions in the lab because I've got that color-coded one, right? Very easy question. Name for the cranial bone that they are next to, right? What's this one here? What's that one there? Yeah, so this is easy. The parietal is one that people miss for some reason every now and then, but for the most part, this is okay. So, got to be able to name all this stuff. I think that we'll do okay with that. What's this thing down here? Cerebellum. All right, we're on top of this. So, two sulci that we're going to have to know, right? There is one, and a sulcus is a groove, right? So it's this groove here that separates the frontal lobe from the parietal lobe. There's another sulcus that's going to separate the temporal lobe from the frontal lobe. Some of the other separations, some of the other divisions between the lobe are just not as clearly defined anatomically, right? There's not a good separation here between the temporal and the parietal, right? Or between the occipital and some of the other ones. So it has more to do with relatively arbitrary assessments, and sometimes it gets tied into functional regions as well. Right? So the two sulci we're going to have to do is this one here and this one here. The two, that's what I just said. Central sulcus, frontal from the parietal. That's a very easy question to ask. Which of the following which of the following grooves separates the frontal from the parietal lobe? And your choices would be longitudinal fissure, transverse fissure, central sulcus, and so on and so forth. So, your book goes into some other lobes, right? I think one of the ones they talk about is if you grab onto this temporal lobe and you retract it, you can see underlying regions of cerebrum. They've called it the insular lobe, although there's a couple of other names for this. I don't have any models upstairs that show that, right? So we're not going to worry overly much about it, right? So really, the ones to know, I'm trying to keep us paired up as much as possible with the lab, because I've heard that somebody has made the final and the lab practical on the same day. Right? That seems unfair to me, but it is what it is. So I'm going to try to keep it relatively similar from here on out. So some things that you may or may not know. A lot of the information coming to and from the brain is going to crisscross on its way to and from. A lot of that crisscrossing is actually happening in the spinal cord, right? But there's some crisscrossing that happens in the brain as well. Right? Have you all heard that your right hand is actually controlled by the left side of your brain? Right? So this shouldn't be terribly surprising. This crisscrossing is called decussation. It's not entirely clear why this occurs, right? What the benefit of decussation is. I've seen some arguments, but never anything really convincing. It's known that most vertebrates do decussate. It's known that most invertebrates don't, right? But the functional significance is not clear.
Don't we actually see upside down when our brain flips it?
Well, that's another issue entirely, because it's when you talk about a lens, that has to do with the physics of lenses and how our lens is constructed as inverting the picture as, right, so they come in. So the short answer is yes. We'll deal with that more when we get to optics, when we do this stuff next week. So there's this whole thing here, right? Some of you can tell, right, who's right-handed, right? Chances are y'all are doing most of your major cognitive functions on the left side, right? And you're leaving the other stuff to the right side. Right? So that has to do with this lateralization, right? Lots of things are going to be done on one side and not the other. So a big example of this, and the one to really make sure that you know for both the lab and for the lecture, but more for the lecture, is a lot of your speech functions are lateralized. Right? So that comes into this phenomenon of dominance. Right? Some things are done on one side, some things are done on the other. Right? The dominant hemisphere does a lot of your language functions. Right? And so they are lateralized to the dominant hemisphere. If you are right-handed, right, then most right-handed individuals are left hemisphere dominant, and so their speech functions are left lateralized. Okay? Do I have any southpaws in here? A lot more than my other class, right? About a 50-50 split in terms of whether you are left hemisphere dominant or right hemisphere dominant. So a lot of your communication, a lot of your higher order functionings are lateralized to your dominant hemisphere, and most people that's the left. Lots of the other things are going to be done on the other side. Right? Now, there's a significance to this, right? And it's that since we're lateralizing stuff, sometimes you need communication between the two hemispheres. Most of that communication gets rounded through a bunch of myelinated axons that are found within the corpus callosum, right? So this is a mid-sagittal cut. The corpus callosum is going to carry information to or from the left hemisphere. You will have the opportunity to do a coronal section within the brain so that you get something that looks like this. What's this here? That's the corpus callosum. And it's literally running information back and forth between the left and the right hemispheres of the cerebrum. So, corpus callosum, conduit information from one side and the other. In individuals who have severe epilepsy, right, that is recalcitrant to other forms of treatment, meaning other treatments are not helping, one of the things that they will consider doing is they will go in and they will slice through this corpus callosum and sever the major connection between one hemisphere and the other. Both of the hemispheres will continue to work, but their ability to communicate with each other will be impeded, right? The thought here is that when you have that electrical disturbance in this pathology, that electrical disturbance cannot spread to the other hemisphere. And so the seizure will not completely take over them and incapacitate them and can reduce the severity of the disease, right? It has some strange side effects, though, right? So, for example, my speech functions are left lateralized, right? And this is the side that controls the right side of my body, right? My sensations and motor functions on my right side are on the same side that my speech functions are. And so in people who have a recent corpus callosotomy, if you have them look away or close their eyes and you hand them something in their left hand, they'll know what it is based on the sensations, but they will not be able to articulate it. They won't be able to give the name for what it is that they are holding. Right? It also can cause some strange identity issues in which actions that are performed by the dominant hand are seen as actions that are under the control of you, whereas the non-dominant hand will seem to have a mind of its own. It's called alien hand syndrome, right? Very bizarre phenomenon, right? It's strange sometimes because we have this tendency to think of ourselves as just being us, right? We are this immutable thing. But we are constructions of our physiology, and large amounts of that is just straight up physiology of the brain. And if you start messing with somebody's brain, you change in a very real way who that person is. Right? So, usually, they go away. Usually. Right? There are other conduits of information that go from left to right and right to left, and sometimes it will reroute traffic through those other regions. Right? So, very strange. All right, so let's go a little more involved, right? We did this in the lab upstairs as well, so this shouldn't be new. We have a model that looks something like this, right? Everybody see those colored models upstairs last time? All right. We're going to start right here, right? We're now going to have to do the two gyri, right? The two gyri, you've got to know my name on either side of this central sulcus. You can call them either by their actual anatomical name, which is the precentral gyrus or the postcentral gyrus, right? Named for their location relative to the central sulcus. So this is the precentral gyrus. It's part of the frontal lobe. This is the postcentral gyrus. It's part of the parietal lobe. or you can call them by their functional name, right? This precentral gyrus is the primary motor cortex. This is the one that's controlling the actions of your voluntary skeletal muscles, right? Almost every single action of a voluntary skeletal muscle is receiving that action potential from this gyrus right here. And you can map it out on the gyrus, and I think your book does that, right? So your primary motor cortex, part of your frontal lobe, voluntary skeletal muscle movement, right? You can map it out. This is a homunculus. It's a representation of a person. The size of the person, or the size of the part of the person, is related to how much brain space is actually controlling movement of that particular part. Okay? What you can see is that you've got most of your brain space dedicated to two relatively small parts of your body, your hands and your face. That shouldn't be terribly surprising. A lot of very fine, very coordinated and precise movements that we can accomplish with our hands, right? This is our way to interact with most of the world around us. A lot of architecture controlling the muscles of the face, right? Most of this is because of speech and vocalization patterns and because of nonverbal communication, right? A lot of communication that you do without saying anything at all. It's just your face. It's one of the first ways that you learn to communicate. Babies very quickly learn how to tell whether you are angry or happy just by looking at your face. One of the most important things for the cognitive development of infants is to look at you in their right, to get facial recognition, to look them in the face. This is a concern. There was a study out just recently that said that this is being interrupted nowadays because parents are spending so much time doing this rather than actually looking and interacting visibly with their children, right? And that's going to cause issues down the line, I guarantee it. So, very important. What's up? I heard something? No? Alright. We got a specialized motor region up here, near the frontal and the temporal, next to the primary motor cortex. This is Broca's area. This is about speech. So a lot of the really sophisticated movements that are required for vocalization are controlled in part by both of these structures, and Broca's region plays a huge role. Right? As human beings, our vocalization patterns are much more complex than even our most closely related primate ancestors. Right? We have developed this ability to communicate verbally much greater than other primate mammals. So we have a relatively large chunk of brain space responsible for generating those articulations. In the lecture exam, this is one of those things that's usually lateralized to the left hemisphere. The model upstairs, though, one of the two at least, has it on both sides, and that's not technically correct. All right, so primary motor cortex, Broca's area, which is speech motor movements. This is the central sulcus again. Going posterior to the central sulcus is the post-central gyrus, also known as the primary sensory cortex, or also as the primary somatosensory cortex, because this is not any sense. This is most of the senses that are associated with the integument, right? This is not the special senses of the head that include hearing and vision and taste and olfaction and all of that stuff. So, part of the parietal lobe, primary somatosensory cortex, is within the post-central gyrus. This is sensory information, what we'll call general sensory information in a little bit from the integument. You can do a homunculus. Again, our hand, large region of the cortex, right? Because this is where our interaction with the world around us is coming from. Lots of sensory information from the face as well, right? Particularly the lips, surprisingly enough, right? This is really because of sexual activity, right? Most people, not everybody, but most people end up kissing at some point pre-coitus or during coitus. Sometimes even afterwards, but that seems unnecessary, right? But a lot of things that are involved in sexual arousal get a large amount of brain space in this postcentral gyrus, right? Lots of lips, teeth and gums, tongue, genitalia, obviously. It's got this huge prominence here for the genitalia. Relatively little space dedicated to the whole rest of the body, and that's true for the primary as well. You just don't need a lot of involved architecture to do what you need to do with your arms and legs. Hands and face. So, I've got a hanging wide here, right? We've got our primary auditory cortex, not surprisingly, part of the temporal lobe. Right? This is a little overly generous here, but we're not gonna pinch pennies. We've got the primary visual cortex way back at the extreme terminus of the occipital lobe, right? These are both reasonable targets for the practical. I like the visual more than the auditory, though, all things said and done. So, and then we've got association and integration areas, right? This is a fancy word for the part of the brain that actually makes sense of things, right? So, they're going to accomplish a lot of higher order functions. They're going to give significance to sensory information, right? They're going to do lots of things that you really associate with the brain, right? Your actual thinking about things. They're usually next to the primary cortex that they are associated with, right? You rub your hand on something soft, like a cat, right? That's going to trigger pressure receptors, right? But it's this part of your brain that says, hey, that's nice. Right? And that's what gives you some of the gratification from petting an animal, right? I think the thing that I did in the lab is I walked up and I threatened to flick somebody in the forehead, right? You can feel the pressure. They always get a little nervous because they never really know whether I'm going to do it or not. Right? Yeah. No, I can't do that. It probably constitutes assault. Right? It describes significance. There's a association area that's next to the visual. This is the visual association area. What is the significance of what it is you're seeing? There's an auditory association that's a large chunk of the temporal lobe. Or basically figuring out what it all means and what is the significance of all of this stuff. Right? There's a very special association area that is right next to the primary cortex for sensation. That's part of the association area. That's part of the auditory association area. That's actually surprisingly close to a lot of the visual architecture as well. Right? This is where we are interpreting the significance of a lot of language functions, right? This is our ability to understand communication, written and verbal. Right? So, usually left lateralized as well, right? Speech and language functions. Not the motor component, but the actual cognitive understanding of speech and language. All right. And then we got the big one, the prefrontal cortex. This is where your thinking gets done, right? This is where your problem solving and your critical thinking is occurring, right? It accomplishes things like planning as well, right? Your ability to predict the future and understand what's going to happen as a result of today's action, right? That's all prefrontal cortex. A lot of other things get affected by this, right? Because you are able to understand that you currently are getting a low grade in my class, and you are able to predict the outcome for that as far as your entrance into the nursing program, right? You are able to become anxious about what's going to happen in the future, right? A lot of good things come out of the prefrontal cortex, but our ability to predict the future is not without consequences as well. Lots of anxiety and stress as a result of actions within the prefrontal cortex, right? Does that kind of make sense?
Is that what ADHD affects?
Honestly, I'm not even going to pretend to know the answer to that. So there are a lot of, I had somebody ask not about ADHD, but about dyslexia and the role of Wernicke's. I just don't have the pathology background to really give you a feel for that. I can look it up if you're interested in knowing, and I may be able to come up with some concrete information. Great question. It is just beyond my own personal knowledge. So, most people don't like anxiety and stress, right? That's actually good for you, though, because it's a motivator to action, right? It should be motivating you to start studying this crap before the exam rolls around, right? So, this was one of our first big treatments for dealing with psychological diseases, right? This is not in the slides. Have y'all heard of a frontal lobotomy before? Right? The idea here seems at least understandable. If this part of your brain right up here is controlling your stress and anxiety, and you want to limit stress and anxiety, were you to sever its connection to the rest of the brain, right, that might solve your problems. So, that's how you do it. That's from an instruction manual for how to perform a frontal lobotomy. You bore a hole in the side of the bone. You take a metal rod and you jam it into the brain. And then, as it indicates, you wiggle it back and forth. In the process, severing the axons that connect the front of the brain to the rest of it. That seems a little barbaric to me, right? I'm okay with the stress when you get right down to it. It seemed a little barbaric to them. As it turns out, punching a hole into your brain through the side of your skull has other complications like infections and stuff like that. So they worked out a more humane way to do this. Right, are we ready for this?
Medication.
No.
Oh, shot.
No, not ECT either. What they do is they push the eye out of the way a little bit. And then they take something that looks a lot like an ice pick and they jam it through one of the fissures in the back of the orbit. That's an actual transorbital lobotomy going on there. So,
Yeah, that looks safe.
yeah, that's kind of gross. The good result, right? Keep in mind this is the good result, is that you don't really care about anything, right? You don't care about the fact that you have whatever mental disease it is they're trying to treat, but you also don't care about such niceties as urinating in the toilet and not in your bed, right? Or all of those other things that seem important to you now, right? That's the good result. So, that was a Nobel Prize for the guy who came up with this, right? So, science is good though, right? It seems kind of shocking, right? One, it just seems horribly barbaric. There was a case, actually, a very famous case, of a mother doing this to their stepchild because the stepchild had a tendency to act up, right? So, but you have to understand some history here. Prior to this, we had no way of dealing with mental disease. There was nothing we could do. What you did with somebody who was mentally diseased back then, and this is relatively recent, right, was you locked them in a sanitarium or an insane asylum for the rest of their life. And they were horrible conditions, and horrible things were done to these people by staff and by other inmates of the insane asylum. This was the first time anything had ever been found that seemed to cure mental disease. And that's why it was received like this, right? That's why. Now, it is horribly barbaric. Right? And there are other options now. Now we medicate in one shape or form. Right? Whether or not we are medicating justifiably in all cases, whether our medicines are doing what they should be, right? That's another issue. But this was the first thing that had ever been shown to work. Okay? So, brainstem. Moving on. So we're top, now we're moving all the way down, right, to the base of the skull, and we're going to work our way back up. Brainstem, 3 pieces. Medulla oblongata, pons, and ligula. Right? These are absolutely essential. Right? You can jam a rod into somebody's brain, into their cerebrum, and wiggle it around, and they live to spell, so they live to tell another day. Right? They live to see another day, tell another day. I'm mixing up my metaphors, as it turns out. Right? So you can do without some of the functions of the cerebrum. My brother-in-law took a gun and he held it to the side of his temple and he pulled the trigger. And he lived, amazingly enough. Right? Because the cerebrum is largely dispensable. It gives a lot of things that are very good for the quality of your life, but it's not essential to maintain basic functions of the human body. Had he pointed the gun in a different direction and it done damage to one of these structures, though, that will kill you.
Is that why putting it in your mouth is much more effective?
Yeah, I'm not advocating one mode of suicide versus another. Mortality rates are higher when it
My
goes.
brother-in-law shot himself.
Yeah,
He
no, I
hit
know.
him through the mouth.
Mortality rates are higher when it goes this way because of damage to the brainstem. That's absolutely correct.
That's where all your primitive functions come from.
Yeah, so this is a lot of our autonomic functions, right? And we'll deal with some of these specifically, but this is going to be respiration and heart rate and all of that stuff. So this is also going to act as a great big relay. Anything coming up has got to go through the brainstem. Anything going down has got to go through the brainstem, right? So it's going to act as a great big relay as well. So foramen magnum. This is spine. This immediate chunk up here is medulla oblongata. Okay? So the medulla oblongata is one of the big ones. It is the major regulator of heart rate, of respiration rate. It's a big controller of what's called vasomotion. And we've talked about this. This is the vasoconstriction and vasodilation, making your blood vessels a little bit larger, a little bit smaller in diameter. Right? This stuff is essential, right? And if you can't do this, if you can't maintain heart rate, if you can't maintain respiration, right, you're dead. Right? Some of our cranial nerves that y'all are going to have to know are emanating from this region in here, right? So 8 is basically, which one's 8?
Activated.
No,
Adducens.
this is, no, adducens is 6. This is the vestibulocochlear one. Accessories 11. We got some memorization to do before the exam runs around for sure. So above and this is not all these systems do by any stretch of the imagination. Your book is not very good at naming some of this stuff, so we're going to keep it simple. Pons. This is the cerebellum here. Pons sits right in front of the cerebellum. What's this cavity here? Fourth ventricle, very good. What's this cavity here?
Subrectum.
No. This is the central canal of the spinal cord. That's okay. great big relay, right? It's a huge relay to and from the cerebellum. We're going to deal more with this when we get to the cerebellum. It assists the medulla oblongata in regulating breathing. Very important function. Another chunk of cranial nerves coming out of the pons. 6 is great for identifying the inferior aspect of the pons, right? So when we go upstairs, if you can find the pons, you can find 6. If you can find 6, you can find the pons. Right? So we'll see what we can see. A little above the midbrain, or a little above the pons, is where the midbrain is, right? This is not midbrain here. This is diencephalon, right? So this is the midbrain. What you're going to look for in the dissection is these bumps here. There are going to be 4 of them all together. So the midbrain is another huge relay. This controls alertness and wakefulness, right? This is going to be responsible for waking up the cerebrum as you transition from sleeping to wakefulness. Oftentimes, damage to the midbrain will put you in a permanent coma. The bumps on the back are going to control auditory and visual reflexes, right? It's also going to involve in muscle tone as well. A couple big cranial nerves coming out of the midbrain. 3 is one that you're going to have to know for the practical if you're in my labs. 3 is a big one. So. On the back of the midbrain, you've got the corpora quadrigemina. It's a set of 4 bumps. You can only see here, though, because this is a mid-sagittal section. I have pulled the whole brain stem and diencephalon out of the brain. You are looking at the dorsal view. Is dorsal posterior or anterior?
Posterior.
Posterior. Right. So I've got 1, 2, 3, 4 bumps that are the superior and the inferior colliculi of the corpora quadrigemina. On the practical, you just got to tell me corpora quadrigemina if you're in my lab. So we'll come back to this picture in a little bit for a couple of other things. Right? The superior bumps are involved in your visual reflexes. Right? They're controlling a lot of automatic movements of the eye. Right? If I focus on something and I turn my head, my eyes will automatically stay tracked on whatever I'm focusing on. Right? A lot of that is regulated by the superior colliculi. Lots of other things as well that these guys are going to do. Some we'll touch on when we get to vision, some we won't. It's pretty interesting all the things that your eyes are actually doing without you actually thinking about it. Right? Inferior colliculi are a bunch of auditory reflexes. Right? This sound filtration here. Human beings are actually very good at listening to other people. In a crowded room, it is very easy to filter out like this humming from the air conditioner unit, right? Because our brains automatically tune out anything that is not human voices if you're trying to listen to a human voice. Another big auditory reflex is whenever there's a loud noise, right? There's a tendency for your eyes to look in the direction of the noise. Right? Did you look in the direction of the noise? A little bit? I know, that's another big benefit. All right, so above the midbrain is the diencephalon. It includes 2 major structures that we're going to care about and a couple of things hanging off the hypothalamus as well. Thalamus, hypothalamus. When you do a mid-sagittal cut, you see only a very teeny tiny little bit of the hypothalamus, right? Or of the thalamus rather. You see only this medial wall, and it's usually recessed a little bit because when you're doing that mid-sagittal cut, what space are you cutting through? The 3rd ventricle. So this is the thalamus here. Right? Oh, it's right there. 3rd ventricle. That's less exciting that you knew the answer to that. Sure you did. What ventricle is that? Ah, very good. You knew that one too, though, didn't you? Yeah. 3rd ventricle, 4th ventricle. That's one. Thalamus. Right? You're cutting through that 3rd ventricle, revealing the medial wall of the thalamus. Right? What's the structure down here? The hypothalamus. Right? It's got this little W shape. We're going to have 2 things hanging off the W. Right? The thalamus itself is a major relay for sensory information. Pretty much all of your sensory information except for olfaction is going to be routed at some point through the thalamus to wherever it's going to end up. Olfaction is kind of interesting, and we'll talk more about this when we get to it. But olfaction is interesting because the nerves that detect olfaction, and that's cranial nerve 1, feeds directly into the cerebrum. Cranial nerve 1 is the one that is really the only one that's coming directly off the cerebrum. And its pathways are distributed. It sends information about smell to all different parts of your brain simultaneously. We tend to think of our sense of smell as being our least relevant sense. But in fact, olfaction subtly influences almost everything that we do and think, right? A lot of our opinions of people or things has to do with what it smelled like at the time that we interacted with them. Right? There's been a lot of marketing research into can we influence people's behavior when they are buying stuff by the smells that they are exposed to.
Is that like pheromones?
Yeah. There's a whole nother branch of biology that deals with how smells affect us in terms of pheromones and how that feeds into mating behaviors and such not. We are affected by pheromones. Our ability to become aroused can be influenced by certain chemicals given off by the opposite gender. Right? So this is subtly influencing everything you do in ways that you probably don't even realize at the time and may never realize. So this is the thalamus here, hanging off of the back of the thalamus, not clearly represented here, but it will be obvious on the models and on the brain because it's a different color, is the pineal gland. Right? The pineal gland, right, this is, I've written the thing out again. These are the colliculi, right, that mark the midbrain. This is the pineal gland hanging off of the thalamus. The thalamus, when you do that mid-sagittal cut, all you see is this little teeny tiny part right there. Okay. This structure here, though, is all part of the thalamus, right? So the thalamus is very, very large. What's this space between the thalamus? The third ventricle. The pineal gland is an endocrine gland. We'll probably deal with it at the beginning of AMP1 or the beginning of AMP2, so I'm not going to go into details. But it is the thing that is controlling your circadian rhythm, right? Your sense of when it's nighttime and when it's daytime. For those, what we said in the lab was that for those of you who work nights, right, sometimes even a full 8 hours of sleep before your shift and you're still tired all night long, right? That's because this gland is telling you it is not time for you to be up and active. It takes a toll. Over the years, you have higher rates of morbidity and mortality if you work night shifts over and over and over again. And it takes its toll over the years. The hypothalamus, okay, named for its location below the thalamus. Okay. This is going to have two important connections. This is a very important structure for a number of reasons, and it's going to be connected to two other structures. Right? So one of the major things it's going to do is it is going to bridge the nervous system to the endocrine system. There is going to be a direct wiring of neurons that have their axons passing through the thalamus or through the hypothalamus into this gland here, which is the pituitary gland, otherwise known as the hypophysis. The way it dangles down here and is cradled within this nest, right? I like to think of this as the testicle of the brain, right? So, what bone is this here?
Your front.
Nope.
Loin.
That's the sphenoid, right? Because this is the cell at first. Are you just shouting stuff out, or?
No, I've seen that cavity.
It does, right? Up here would be the front. No, I mean that's not a bad guess. Sometimes I wonder that people don't say things just to get me to keep on moving in the monologue. But that's it. One of these days, I'm just gonna go. I'm gonna do the thing from Ferris Bueller, right? I'm just gonna go. Anybody? 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 The sphenoid? No. Not familiar. Now I totally lost my train of thought. Hypothalamus, pituitary gland. Oh yeah, testicles. Right? It's always about the testicles. Right? It's a gender thing. Right? So, but it works so because they dangle, and the testes are endocrine glands, so it kind of works. You won't forget it now, though, so it's okay. Bridges the nervous system, regulates the pituitary gland. The pituitary gland is the heart of the endocrine system, right? And so that means that the hypothalamus is going to be involved in all the stuff that the pituitary gland regulates, right? So larger roles in homeostasis. Heart rate, blood pressure, temperature, electrolyte balance, hunger, thirst, so on and so forth. My guess, this is going to come up in AMT 2. We've got this other thing hanging off of the hypothalamus, right, which is the optic chiasm, which is part of optic nerve 1, or cranial nerve 1. Now I'm messing it up. The optic chiasm, part of the optic nerve, which is cranial nerve 2. And what's cranial nerve 1? Olfactory. We've got a big set of axons that are connecting the hypothalamus to other parts of the brain. Right? This is the fornix here. These two are going to be big landmarks for you in that dissection. Right? This is the septum pellucidum behind which the lateral ventricle sits. One of the things you will do in the dissection is potentially slice through that septum pellucidum and pull it open to see the lateral ventricle behind. Do you have a question, Brenda? You just sticking your finger near your mouth? Okay. All right. So, again, I pulled this out. This is no longer a dorsal view. This is a ventral view, so you can see the other side of things. Right? What's this region of the brain here? No.
No, this is on the wrong side.
This is the medulla. Pons is up there. Okay. This is the optic chiasm that's hanging off of the hypothalamus. This is the pituitary gland here. These are some structures that you're not actually going to be looking for if you're in my lab, but they may be a little confusing. These are the mammillary bodies there. All right, so that's cranial nerve 2. What's this one here? 3. What's that one? What's that one? No.
6.
Yeah, that's 6. That's 5 there.
Oh, you got me.
I know, right? Fornix, hypothalamus to the cerebrum. That's going to allow us to participate in this larger group of structures known as the limbic system if you have to deal with that in psychology at some point. Pituitary gland hangs off of the hypothalamus by the infundibulum. We won't actually see the pituitary glands in those brains upstairs because the meninges have been removed and that rips off the pituitary gland as well. But we'll be able to see the location that it was connected. The pituitary gland is sheltered within this feature that we learned previously. This might be a good one to explain. The cerebellum. This is an interesting one. Posterior to the brainstem, posterior to the fourth ventricle, in fact. Inferior to the cerebrum. Looks like a little bitty mini brain with its architecture of gray and white matter. So, white matter is the arbor vitae, gray matter is the cerebellar cortex. It's separated from the cerebrum by this enfolding of the dura mater called the tentorium cerebelli. Okay. There is also a split within the cerebellum itself called the falx cerebelli. You will not be able to see this in the lab upstairs, though. So, in all likelihood, I'm just going to remove this from the future. I think I'm not going to talk about this anymore. So this is not going to be on the lecture exam. That one might show up though. Gray matter. What's in the gray matter? Yeah. Yeah. Cell bodies of the neuron. Unmyelinated axon. This is myelinated axon. There is a lot of communication to and from the cerebellum. You've got these great big connections that are called peduncles, right? Kind of a strange word. We're going to bring information into the cerebellum through 2 different peduncles. They're going to bring sensory information in from the body, right? For example, telling me exactly where my hand is. It's going to bring information in through another one of the peduncles from the cerebrum that's going to say where I want my hand to be, right? And then the cerebellum is going to compare those two things. And it's going to say, hey, that's close, but not quite. And it's going to send out instructions back to the cerebrum, ultimately down to other parts, saying, this is what you need to do in order to make your desires match your current circumstances, right? So what the cerebellum is doing is a lot of course correction. When you are reaching out and trying to grab onto something, your cerebellum is constantly monitoring that process and sending out thousands of little course corrections along the way. Very, very important for fine motor function. People with damage to the cerebellum often lose a lot of their motor coordination as a result because they can't do this constant compare and contrast. So one of the main roles of the cerebellum is just these constant motor adjustments to make sure that what you want to do is what you are actually doing. If you do that same thing enough times, what your cerebellum will do is automate that process, right? Now, I asked my first class who was able to touch type, but nobody really understood what I meant by that. Who is able to use a keyboard without actually looking at the keys? Right? That's not very many people in here. All right, let me do this. Who is able to, on their phone, text really fast without having to devote a lot of energy in your brain to it? Yeah, it's a little bit more. I know some of the people up here because I've seen you do it. Right? So, now, have you ever experienced a phenomenon where normally you don't even think about it, you just do it, just or like this, right? And you don't even think about it, and it works perfectly. And then you stop for a second and you start thinking about what you're actually doing, and then all of a sudden you start making loads of mistakes where you never would have before. Has any of you experienced that before? That's because all of a sudden you are not doing this automated program that was dictated by your cerebellum anymore. Once you start actively thinking about it, you are not as good at doing that task, right? So the other function of the cerebellum is to develop this automation, this procedural memory. Sometimes it's this colloquialism, your muscle memory, right? In which something that you've done thousands and thousands of times, your cerebellum comes up with this automated program that just does it for you so that it doesn't require your conscious thought. And it works much, much more efficiently than your cerebrum trying to run the show itself. This is a big part of development for little babies, right? This usage of the cerebellum, this initial course correction that they do, and this initial programming, right? And you can tell that little babies can't do it yet. They haven't developed these programs. Who's got little kids in here or have them at some point, right? When my experience with the little infants was they see something they really want and they get all excited and they start cooing and they start drooling all over themselves. And they reach out and it's like they're morons because it's right here, but right over there, right? Nowhere near it. And you're just like, God, really? Do I need to take this kid to the doctor? Right? But it's just because they haven't developed this ability to coordinate things yet. And eventually they finally get the hang of it and they reach out and they grab it. But they don't grab it like you and I would. They don't grab it with just a couple fingers. They take the whole thing in their fist, right? Because again, their cerebellum hasn't gotten the hang of all of this stuff yet. So, all right, so we're through the brain. Right? This is easy. We've already done this in the lab, and I'm not going to belabor this. One of the things, right, but I am going to assume that you already know it, right? And so if you don't, you need to let me know. We'll come back and do a little bit more with the spinal cord on Tuesday because we're going to get into the peripheral nervous system on Tuesday, which means we're going to be dealing with nerves. You got to know what all this stuff is, though. Right? Gray matter versus white matter. be able to compare the arrangement, the anatomy to the cerebrum and the cerebellum. Are we okay with this? Y'all did really great for those of you who are in my lab. Y'all did really great with those labelings for the most part. Are we okay with this stuff? Any questions? All right. So this we already did. Not any different in the spinal cord, right? We're going to start talking nerves, right? We're talking cranial nerves already in the lab. Nerves are always peripheral nervous system, right? So if we're bundling up axons together in the peripheral nervous system, we're going to call it a nerve. You would never refer to something in the central nervous system as a nerve. You always call it tracts. This term we're not going to use as much. In your lab notebook and in your lecture book, there is a whole collection of random tracts within the spinal cord. They go up and down the spinal cord carrying information to and from. We're not going to have any of that on the test. There's no clear delineation of that stuff in terms of anatomy. You can only tell those tracts by the physiology, by actually watching the signals going up and down. This is a term that we're just not going to get a ton of use of. But I want you to know that if I call it a tract, it's not peripheral nervous system, it's central nervous system. Right? And that's about as in-depth as we're going to get. A bundle of tracts is a funiculus, and you do have to know a couple of the funiculi. Right? Although our bundling is not going to be as clear as it is here because we're not actually wrapping anything in dense, irregular connective tissue like we do in the peripheral nervous system. So we're just not going to focus on this as much. This we're going to do in detail on Tuesday. We're going to close on the reflexes. And I've just gotten lazy and I'm using your book slides, and I apologize for that. There's just a couple of them though. So reflexes, I think most people are able to define at least somewhat coherently what a reflex is. It's an automated action, right? That you don't think about, that you're not really consciously directing. It's just an automated program, right? You take a rubber mallet, you bang on that patellar ligament, and that leg's going to go up like that, right? Just as an automated response of an otherwise voluntary set of muscles known as your quadriceps. So, information comes into the central nervous system, action goes right back out. Right? This is called a reflex arc. The reflex arcs that we're going to talk about are spinal reflex arcs. Right? Your brain is not involved in spinal reflexes. Information is going to come in from a sensory neuron, which is a unipolar neuron whose body is in the dorsal root ganglion. That information is going to come into the central nervous system. Here, it's shown as having an intermediary central nervous system neuron called an interneuron. Right? Interneurons are always central nervous system, never peripheral nervous system. And then information is going right back out again through the motor neuron, right? And it's all in one step. Action potential here, action potential right back out, right? It's very quick, but it's not very flexible. It's only going to do one thing every single time you generate an action potential here, right? Y'all follow me so far? This is one of the simplest examples. You bang on that patellar ligament, and I've got mallets upstairs. If anybody really wants to hit their lab partners with a rubber mallet, that's fine. Do it in the patellar ligament, right? I had one semester somebody banging on somebody's kneecap. That's not going to do anything other than make it hurt, right? Certainly don't bang anywhere other than the patellar ligament. Just two neurons in the simplest examples of spinal reflexes. One sensory neuron, one motor neuron. Information comes in, information goes right back out again. Right? Very simple. Right? It's this one we've already seen before. A slightly more complicated variation of this is something called the withdrawal reflex, right? You reach out and you touch something that's unexpectedly hot. What do? Jerk your hand back. Jerk your hand back before your brain has even told you, hey, bye-bye, that's hot. You may not want to do that, right? The advantage here is that it's fast, right? Because the signal doesn't have to come all the way up and go all the way back down again, right? You touch it, let go, right? This is one that I experience a lot too, right? I'll come out of my room in the middle of the night because the damn cat won't shut up. And my 5-year-old, I guess they're 6-year-old now, my 6-year-old, however the hell old my kids are, has left a cloud of debris all over my floor. And I will step on one of their toys. Yeah, Legos are horrible. Horrible. And about 90% of the time I will manage not to break the offending piece of junk on the floor because I will jerk my foot up, right? That's another kind of withdrawal reflex. Information comes in, it's bridged by the central nervous system interneuron, comes right back out again, and it causes a contraction of my, for example, biceps femoris or semitenosis that causes a flexion of the knee, right, and of the hip. This is 3 neurons, a little bit more complicated, but still it's okay, right? They get much, much more complicated, right? As I am lifting this leg up, it's not a real good time to be flexing my other leg as well, right? Because that'll put me flat on my posterior in the middle of the night, and I'm already mad to begin with, right? So there's something called a cross-extensor reflex. At the same time you're causing this one to flex, you want to bridge over to the other side, send out an action potential causing this one to contract and this one to relax, right? So that you don't fall over in the process, right? So these become more complicated. Now, at the same time that you're initiating these reflex arcs, you're also sending information up the spinal cord to the brain saying, hey, by the by, something going on down there. You may want to figure out what it is and do something about it, right? And then you start to feel the pain of stepping on that Lego in the middle of the night. And you can catch yourself before you completely overtop and overbalance, right? So this information does get routed to the brain eventually, but the immediacy of the response requires that brain not be involved, right? Automated response. Lots and lots of reflexes. You will deal with some of them as a clinician potentially to see whether the nervous system is functioning correctly. One of the ones they do with little babies is called the Babinski reflex, where they rub something on the foot and see what way the toes curl, right? You can override these reflexes. What they do when you're doing the patellar reflex is they say you gotta relax your leg. With enough conscious volition, with enough willpower, right, you can keep the reflex from happening, right? If you contract these muscles when they start banging on that ligament, your leg's not going anywhere, right? You can override these reflexes. That pot of water, if you see that pot of water getting ready to fall on your kid because the kid reaches up for the 57th time and grabs onto it even though you've told him not to do it, you're going to grab that burning hot pot of water and keep it from falling on your kid. Right? You can override these reflexes with voluntary action. Right? But without that voluntary impetus, right, these reflexes are going to go on. So that's all I've got for you today. We're right on time.